Cosmic Plasmids were developed on the late 1970s in response to a need for vectors that are capable of accommodating large segments of genomic DNA. It contains a lambda-phase COS sequence that facilitates in vitro packaging of genetic materials. It can accommodate around 31-44 kilobases of foreign DNA. However, it is not suitable for mammalian genome and also its efficiency is low. Bacterial artificial chromosome or BAC. It is a high capacity vector. It can contain around 100 to 300 kilobases of foreign DNA. Because of its high capacity, it can transfer the gene's specific promoters, any regulatory domains of both 5' and 3' region of a gene to check the tissue-specific activity of a gene or a protein. It is based on a fertility or F factor of E. coli. F factor allows genes to be transferred from one cell to another through conjugation. It is easy to separate and is a powerful tool of mapping and sequencing of mammalian genome. So yeast artificial chromosome. So yeast artificial chromosome vectors were come into play in molecular cloning from 1987. It can ligate large fragments of genomic DNA. It is also a high capacity vector. It can contain around 250 to 400 kilobases of DNA. It contains an autonomously replicating sequence that is the origin of replication of yeast. They were replicated and inherited by progeny without integrating into the host chromosome. That is also an extrachromosomal entity. It is a powerful tool for mapping and sequencing for mammalian genome. Then expression vectors. It is a basic tool for biotech industry. It is used mainly for the gene expression and protein production. Because of this vector must contain a promoter, transcription, and translation initiation, and also a termination sequence of a protein. So shuttle vectors. Shuttle vectors can propagate in two different host species, like yeast shuttle vector. The advantage of shuttle vector is the DNA inserted into the vector can be tested in two different cell types and also can be tested in a prokaryotes as well as in eukaryotes. Normally, in the diagram, you can see a shuttle vector of E. coli and yeast cell. For the E. coli, it has the origin of replication of bacteria plus a selectable marker, which will be used to detect the vector into a bacteria. On the other hand, it also has a yeast components like a yeast origin of replications and a centromere and a selectable marker, which can be detected or selected in yeast organism.